Alors, merci beaucoup à tous et bonjour à tous ici et Only. Et je suis content maintenant et merci beaucoup pour committee pour inviter moi présenter mon chercheur aujourd'hui. So today I am Nunthawan Nowwarote. I'm working in the team of Professor Valerie Kamangde and Professor Aryan Berdal at Imagine. So today I would like to present my work in the topic of analysis of human craniofacial phenotype in FAM20A mutation from clinical to cellular perspective in the enamel-renal syndrome or ERS. So what is FAM20? FAM20 family is the family of secreted protein kinase that play crucial role in the biomineralization, particularly in the formation and regulation of mineralization. As you can see in the figure, they show that FAM20A are working as a puzzle kinase, so they need to activate the collaboration with the FAM20C to have the function of mineralization, including in the nutrition, biomineralization, serum plasma, and also in the heart. Whereas FAM20B, they are working different. They are working in the function of proteoglycan biosynthesis. And we have been found that FAM20A mutation are present in the patient who have enamel-renal syndrome. And Professor Muriel in our hospital, they have the extensive diagnosis of the patient who have this syndrome, which is the diagnosis criteria including gingival hyperplasia, amylogenesis imperfecta, and renal calcification. And also, as you can see in the soft tissue that you can see here in the gingival and follicular sac, they are also show the ectopic mineralization. And moreover, we would like to understand how ERS in the molecular level. So we would like to translate our research from the clinical research into clinical research and how we can use to treatment in our patient. So at the first time, we will use our patient from the cohort follow-up and also the tissue biome banking. And this project is initially based on the cohort with the identify of FAM20A sequence at the center of the reference for oral and dental rare disease at Losch's Hospital. And we use the biobanking of oral cell to correct our surgical tissue from the hospital. And here is the mutation of FAM20A genes as demonstrated or the identification mutation. Since update 2020, we found that all the cases have been published for the 70 cases and affect in 50 family. And the total of mutation that have been found is 100 mutation allele. The most of the patient that they found the allele, a lot of mutation they found in the patient are Brazilian and following with the Turkish and also in Thai. And how about in France? We found that our population in 22 patients from the 60 family, we found 12 mutation allele, and we found 4 cases that we are unpublished, and we found 3 new mutations that have been shown in the red color, that there are 1 homogeneous and 3 heterogeneous. And moreover, in the clinical investigation has been investigated by our PhD student Sabah Sahi and Professor Muriel at the Losch's Hospital by using X-ray film of the patient. And we are follow up the treatment before and after treatment of the surgery. We give the score to the of the teeth by the 0 for the tooth, erupted tooth, the 1 for subjunctive inclusion, the number 2 for superficial impact tooth, and the number 3 for the deep impact tooth. And our preliminary data result, we have been shown that all the ERS patients have been shown significantly unerupted teeth comparing to the patient in the normal patient at 12 years old. And following with the mutation, as you can see in the different color, are showing the different family of the mutation. And in the line show for the homogeneous patient, and in the dot line show for the heterogeneous patient. As you can see, in the STAR, we follow up the patient who have to visit the clinic more than 3 times. And as you can see, our results show the diversity of the mutation in the ERS patient. And as you can see in the line start to go to the erupted tooth, which is we can see that the significantly when we follow up the patient who come to visit the clinic, so we can predict and we can give the suggestion to the patient who have this syndrome to come to visit to the clinic early as that they can do. And moreover, in the qualitative and quantitative analysis of craniofacial profile have been investigated within our team from the clinical by Professor Lia, Dr. Bridges, and Professor Muriel. So they use cephalomatic analysis of the profile and frontal radiograph evaluate craniofacial feature. And if you can see in the red line and the blue line, parallel with the black, that line in which is show the facial structure. If the lines are not parallel with those the color, which means they are show abnormal of the jawbone. And the result will show that they show the notable variation in the maxillary deep and increase in the maxillary high. And also in the mandible, there is significantly shorter than the usual. So we saw the phenotype of the patient as a class 2 malocclusion. And we can suggest that the potential underlying cellular and molecular dysfunction in the bone growth, remodeling, and development linked to the FAM mutation in the craniofacial formation in the ERS patient. Therefore, we would like to investigate in the molecular mechanism. We collect the cell from, alveolar bone cell from the patient who come to visit our hospital more than 3 times follow-up, and she has the patient has the class 2 malocclusion phenotype. And we confirm the reduction of FAM20A expression in the mutant alveolar bone cells. We found that the FAM20A have low reduction in the mRNA expression and also protein expression. If you can see in the cellular part, you can see localization of the FAM20A inside the Golgi apparatus, which is that list show no significance between mutant and normal cells. But you can see only the difference expression from the FAM20A protein. And after that, we would like to see if the alteration of FAM20A are altered to the function of cellular. So we found that the mutant cells remain round and disturb organization of delay cell spreading compared to the control cell from the well-organized attached cytoskeleton structure, and they show spreading in the overtime, which we observe since 30 minutes until 24 hours. And moreover, we found that in the alveolar bone cells from the patient show compromised cell proliferation and also show severe impact colony forming unit ability. And this result, we can conclude that the mutation of alveolar bone cells potential due to its outer lineage commitment as a mature osteoblast, whereas the control cell, they have the characteristic of mesenchyme or progenitor cells. As you can see, the low capacity of the cell are dividing. And moreover, we see the function of the mineralization. We see that the mutant cells decrease the mineralization during osteogenic differentiation by using ALP expression, also alizarin-less staining, von Cossa staining to see the calcium accumulation inside the cells and also outside the cell. We see the mutant cells show the lower mineralization compared to the control cells. And this is, we can conclude that the mutation disturbs the osteogenic differentiation, leading impair the bone formation mineralization in the alveolar bone cells, strongly support it in the craniofacial abnormalities. Moreover, we would like to see is that possible to have some intervention from the ERS mutation alveolar bone cells. So we use Wnt signaling pathway. As everyone know that Wnt signaling pathway is the major pathway which is regulated with the mineralization of the bone. So we confirm that our cells are involved in the Wnt signaling pathway by using Wnt inhibitor processing, IWP, and we found that Wnt signaling pathway involved in the alveolar bone cell mineralization by prevent the translocation of beta-catenin into the nucleus. And moreover, we are very surprising that we can rescue mineralization by using the inhibitor to inhibit Wnt antagonist by using SFRP1 and bio, which can inhibit Wnt antagonist, as you can see, and inhibit. This inhibition can rescue the mineralization of alveolar bone cells. And this is the highlighting the potential therapeutic strategy for the bone defect associated with the Wnt pathway, provide a foundation for the regenerative treatment. But this finding is not just only for the oral craniofacial, but it also can be the treatment for skeleton bone defect as well. And to conclusion, I would like to this information that we got from the clinical can be like the guideline and the guideline for the surgeon can do, and the planning for the patient treatment when they come to visit, and give the information that if they come to visit the clinical as early as they can, it might be easier to give the treatment and can be to help for the patient to have the good oral cavity in the future. And also the result that we found from the molecular level can give the diagnostic or also for the drug treatment for the patient who have low mineralization. And I would like to thank our team with Professor Valerie Kamangde, Professor Ariane Berdal, and also other clinicians in the hospital logis to help our patient to take good care of our patient, and also all my colleagues in our research group too. Thank you very much.
Thank you very much for your talk. I'm sorry, I must have missed a step. What is the link with between FAM20A and the Wnt signaling?
So FAM20A is they are pusokinase. They are working with FAM20C to regulate phosphorylation of the protein, which is including the protein who are important for the mineralization. And Wnt signaling pathway is the major pathway which is regulate bone development and also mineralization as well. That's why we see the relationship between FAM20A and Wnt signaling pathway.
Did you observe a variation of the mineralization depending on the level of the inhibitor that you use?
It's very good question. It's because this one now we are ongoing to observation, because it was like this finding that we found is quite. I found this result just only few days ago. So now we are ongoing to investigate the concentration as you mentioned. Thank you very much.